Thank you, Christian. And what time is it in Santiago?
It's 3:07 p.m.
Okay, very good. Well, good afternoon, everyone. Thank you for the invitation and the opportunity to talk. As you know, it's more than a seminar for me. It's actually visiting with colleagues. When I joined the BUC in about 5 years ago, I was intrigued by the collaboration with the University of Chile and your own aging center. And as you know, and I immediately was interested in actually making these interactions stronger. And I think little by little, I'm finding my footing at the BUC, and I think and these interactions are growing and getting stronger. So today I'm going to be talking about immune aging and NAD metabolism. And the first slide, if it will move right now, it is not, is really to remind you where the aging field. Years ago, Lopez and colleagues published this landmark review on the hallmarks of aging in Cell 2013. And what they were able to do is to identify a number of molecular abnormalities that characterize the aging process. And we're all familiar with mitochondrial dysfunction, genomic instability, cellular senescence, epigenetic alteration, stem cell exhaustion, telomere attrition, modified or altered intercellular communication, loss of proteostasis, and deregulated nutrient sensing. So these are a number of characteristics that really occur in all living animals. And while this type of organization has been very helpful in sort of organizing the field, it obviously does not fully recapitulate or explains the complexity of aging. And one thing that is, for example, not understood is what is the hierarchy between all of these hallmarks of aging? How do they interact together? I believe that what the field is working on right now, and will work in the coming years, is really trying to understand what is the most important aspect of all these modifications. And within each of these modifications, how do each of these pathways relate to the others? And the story I'm going to tell you today is a story that started by the study of deregulated nutrient sensing, which has been a big area of focus in my lab. And I hope to show you that by the end, deregulated nutrient sensing, in particular NAD metabolism, is actually affecting many of these other hallmarks of aging. So I think it's obviously one step that my lab is creating, but I can tell you there are many other labs working in the same area. Now, the practical implication of this is that we are entering a phase of aging research where we are devising interventions that target the aging process. It therefore will become really critical to understand of all these modifications and of all the targets within each of the modifications, what is their hierarchy? Which ones should we target together? Are there any synergies between these drugs and modification? And I think this is where the field is. We're moving slowly from a stage where we're implementing one single interventions to actually combining them in different manners. And I think this is really something that excites me about the future of our field. So I think we've entered a phase in aging research where we've identified many of the key players, and now is the time for a deeper integration of all of these mechanisms and their relationships. So the particular area that I told you I had been focused on for many years in my lab is trying to understand how does calorie restriction, which is shown at the top, lead to an increased health span and lifespan in green, and along with these increases to an inhibition or suppression of the chronic diseases of aging. And the area that we have focused on, because of my background in epigenetics, is really trying to understand how does an external modification, such as decreasing the number of calories, modify aging. And so we have identified two levels of network, a so-called metabolic network, which is a series of metabolites that include NAD, acetyl-CoA, S-adenosylmethionine, alpha-ketoglutarate, and beta-hydroxybutyrate. So these are metabolites whose abundance change as a function of nutritional status and as a function of calorie restriction. And the interesting thing is that these metabolites are not just part of intermediary metabolism, they also have very strong signaling properties. They interact with a number of key master regulators. For example, beta-hydroxybutyrate, we've shown is an HDAC inhibitor. There's evidence that alpha-ketoglutarate regulates protein demethylases, histone demethylases. S-adenosyl methionine regulates DNA and protein methylation as a methyl donor. Acetyl-CoA regulates histone acetyltransferases. And today I'm going to be focusing on one single of these interactions, is the relationship between NAD and sirtuins. Again, NAD also fluctuates as a function of nutritional status. And sirtuins are enzymes that have been identified, and some of them are epigenetic regulators, and thereby control many of the characteristics of lifespan and health span. So obviously, a lot of work has been done in this field. We're not the only ones doing this, but one area that particularly interested me is the realization that NAD, and so this NAD sirtuin couple here is really a key area for us to study. And one of the observations that's been made in the last 10 years is the realization that actually NAD levels decrease during aging. And this occurs in multiple tissues. I'm showing you here a summary of a review that was published by Yoshino and colleagues in Cell Metabolism in 2018. And what this review highlighted is the fact NAD levels decrease not only in muscle, liver, adipose tissue, brain, and so on, but also in multiple species. It's been shown in humans, been shown in rats, in mice, and so on. What has really not been clear is the reason why NAD levels decrease. Now, so before getting into this, and which will be the focus of this presentation, I want to review very briefly what we know about NAD metabolism. So there are two major biosynthetic pathways that lead to NAD synthesis that are both driven by the diet in part. One is originates from nicotinic acid, or vitamin B3, and leads to a so-called Price-Handler pathway all the way to NAD in a series of biosynthetic steps. Another way in which you can synthesize NAD is via tryptophan in your diet, and this is via the Kineranian pathway. Now, these two pathways are active in different cells at different type and different times. Vitamin B3 is a critical vitamin. If it's lacking, or if tryptophan is lacking, or nicotinic acid is lacking, a clinical syndrome called pellagra develops, which is associated with a series of really critical manifestations that can lead to death. Now, when we get to NAD itself, NAD has two different types of activity. So why is NAD so important? NAD has two different types of activities. First, it can serve as a hydride acceptor, so that this NAD-NADH couple is utilized by a number of redox enzymes, which rely on NAD for their enzymatic activity. And as you know, there are multiple. So this is one way by which the cells can actually shuttle energy from example, from glycolysis to the mitochondria or within the respiratory chain. So the NAD-NADH couple is very important in terms of really allowing the intermediary metabolism to be functioning efficiently. Now, in addition to this, there's a number of enzymes that actually use NAD, but they do more than use it, they consume it. And so this is what I highlighted here in the blue box. These NAD consuming enzymes include sirtuins. We've mentioned this very briefly. They include poly-ADP ribose polymerase. There are many of them involved in DNA repair. Involve also other enzymes, such as a number of mono-ADP ribosyl transferases. Involves CD38, I will be discussing quite a bit today, and CD157, which is closely related to CD38, but much less studied. So in addition, the degradation of NAD by these NAD consuming enzymes generates a product called nicotinamide, which is shown here. And this causes a problem for the cell because meaning constantly the NAD is being decreased. And so there's a so-called salvage pathway, which is highlighted here, which is comprised of two enzymes, NAMPT, and a series of three enzymes called NMNATs. I won't spell out their names. There's three of them that are present in different parts of the cells. There's a mitochondrial one, a cytoplasmic, and a nuclear one. And each of these two enzymes actually take the nicotinamides and reactivate it into NAD. Now, so obviously, if there's an NAD decrease during aging, you can imagine a blockage in any of these pathways could potentially play a role. And so there's been a lot of interest in trying to understand how does it happen. And our hypothesis that we formulated in a review that was published in Science in 2016 was that these NAD consuming enzymes are actually in competition with each other for a restricted pool of NAD. And so if one of these enzymes becomes dominant, you can imagine how it's going to be pulling NAD away from the others. And in this case, I've shown two of the enzymes that people have thought might be playing such a negative role, which would be the PARPs or CD38, CD157. So imagine circumstances, for example, for PARPs, we know that during aging, shown here at the bottom left, there's increased DNA damage. It's been documented by a number of laboratories. This increased DNA damage, we know, activates PARPs. And you can imagine that we know actually in a number of cells, PARPs activity actually increases the function of aging. The best example of this is actually a series of human clinical syndrome, cocaine syndrome, xeroderma pigmentosum, XP, or AT stands for ataxia telangiectasia. So these are three DNA damage repair problems. People do not repair their DNA. The DNA damage persists. These patients actually show accelerated aging and a shortened lifespan. And therefore, they're almost like a caricature of a chronic DNA damage. And we know in these cases, the PARPs are chronically activated, and these patients show very low level of NAD and an accelerated aging. And there's actually current effort to try to treat or to alleviate part of their symptoms by restoring NAD levels. So it's clear for those, but it's a little less clear what is the relative role for PARPs in this process. Today, I'm going to be focusing a bit on CD38 because we think at least some of the evidence that we have accumulated seems to indicate that it is one of the major players. And so what is CD38? It is a nectoenzyme. Actually, it's found mostly on immune cells, CD4 and CD8 T cells, on B cells, and NK cells. It's shown here as with the CD38 catalytic domain facing outside of the cells. So it takes NAD, generates nicotinamide and ADP ribose. It also generates cyclic ADP ribose, which is a cyclized version of ADP ribose, which has some signaling properties. Now, one thing we have learned is that depending on the cell type, the proportion of CD38 that is outward facing versus inward facing is different. I'll show you some data on macrophages, for example, where close to 40% of the protein is facing inward. In T cells, it seems to be about 80 to 90% facing outward. So this has important implications because I'm going to be discussing some of the potential consequences of NAD levels, not only outside of the cell, but also within the cell. Now, CD38 has been long known by immunologists. It functions in cell adhesion, in signal transduction, and in calcium signaling via the generation of this cyclic ADP ribose product. And interestingly, multiple myeloma, this is a cancer which is derived from B cells, express a very high level of CD38, and this has been used as a therapeutic target. There's a company commercializing a Darzalex or daratumumab, which is a monoclonal antibody against CD38 that allows you to kill selectively the cells that express a lot of CD38. So one reason why we got into the field, into this area that I'm going to be discussing, is a paper that came out in Cell Metabolism by Eduardo Cini and colleagues at Mayo Clinic in 2016. And what they showed is they were interested in trying to find what is the mechanism of CD38 and other proteins in the decrease of NAD levels. And what they showed was that if you compare mice of different age, from 5 to 32 months, so 32 months for a mouse would be quite old. Average maximum lifespan in mice is about 36 to 38 months, at least in our facility. And they were able to show that CD38 expression actually increases pretty strongly during aging, as shown on the top panel. And this occurred not only in adipose tissue, but it occurred also in many other tissues that they looked at in their paper. What drew our attention was the realization that when they looked, and so in that adipose tissue, they showed, for example, that the NAD levels progressively decrease during aging. And remarkably, when they looked at NAD levels in a CD38 knockout, they were able to show that these mice actually were protected against the age-associated NAD decrease. And so this really implicated CD38 as the major driver. And without going through the paper again, the same exact data was shown not only for adipose tissue, but it was shown for the heart, it was shown in the brain, in muscle, and in multiple tissue, really painting a picture of CD38 as a key driver of this NAD decrease. And so this got us interested. One thing that was not addressed in the paper, and as an immunologist that really triggered my interest, is the fact that we knew that CD38 was not expressed in adipose cells. And I thought, they did not really address in which cells this increased CD38 expression occurred. And so I had a new postdoc in the lab, Anthony Covarrubias, who decided to start looking at this. And the first experiment we did is to prepare what we call a stromal vascular fraction, which is essentially the cellular component of adipose tissue that is not adipocytes. And the way you separate it from the adipocyte is you first dissociate the tissue and then you centrifuge it at low speed. And the adipocyte, because of their fat content, have low density, they float to the surface. And so you get a nice pellet with lymphoid cells, endothelial cells, and other non-adipose cells. And we can use flow cytometry using an antibody specific for CD38 to actually try to determine, okay, where's CD38 expression located as a function of age. And what the data showed us when we compared mice that were either 14 months or 25 months is that the blue peak here corresponded to increased CD38 expression in 25-old mice. And you can see it was all in macrophages. There was maybe a very, very small increase in non-macrophage leukocytes, but the bulk of the increase in CD38 occurred in macrophages. And so we've pursued this, trying to understand more about the nature of these macrophages. The first experiment that we did with Anthony was to try to separate the two macrophages. First to count them to see if there was a change in macrophage numbers. And indeed, as a function of aging, and this was actually quite surprising at the time, we saw a progressive increase in the number of macrophages as a percent of the total stromal vascular fraction. You can see there's close to a threefold increase in macrophage number. Now, there are two different kinds of macrophages in adipose tissue. There's what we call the resident macrophages, which are characterized by this absence of this marker, CD11C, and there's the so-called non-resident macrophages, which are CD11C positive. Now, when you think about resident versus not resident macrophages, you might not have heard about those in the case of adipose tissue, but I'm sure many of you are familiar with the idea that microglia, for example, in the brain is a resident macrophage of the brain. The Kupffer cells in the liver are the resident macrophages of the liver, and so on for pretty much every single tissue has a set of macrophages that are deposited shortly in utero or shortly after birth, and do not circulate. The non-resident macrophages are bone marrow derived, and they tend to circulate and to go into different tissues to actually perform different functions. And what was quite interesting here is the fact that we found that most of the resident macrophages were increasing. You can see this is where the total number went from about 2% of the total SVF to close to 6% of the total SVF. And the non-resident macrophages actually did not increase. I think there is Meili who is making a lot of noise. So the next question that we ask is, in which of these a subset of macrophages? So this is an increase in total number. The next question we ask is that when we looked at individual macrophages, what was CD38 expression? And so what we saw is that, again, when we looked at the CD38 high macrophages, they were mostly in the resident macrophage population, as shown here on the bottom left. And the CD38 high cells actually contained almost no non-resident macrophages. So this really was quite interesting, highlighting a novel population, and triggered us to continue to try to understand what was happening there. So just a way of introducing the whole macrophage field to tell you a little bit more about them. As I mentioned, there are resident, non-resident macrophages. Despite their different origin, at the end, they present many of the same characteristics. There are two broad classes of macrophages, and this is, I have to say, to highlight that this is a bit of a caricature. There's the so-called M1 macrophages and the so-called M2. We now know that these opposite macrophages have very different functions, and there are many different intermediate forms of macrophages between the M1 and M2. The M1s are classically activated by a bacterial product, LPS, and they're known to be involved in aging. The whole idea of this inflammaging, it corresponds to an increase in these pro-inflammatory macrophages. They're known also to respond to pathogen, and they are thought to be implicated in the type 2 diabetes development. The M2 macrophages are activated in a different way, predominantly by two cytokines, IL-4 and IL-13, which are secreted by the so-called Th2 lymphocytes. And this is where the M2 comes from. And these macrophages actually play a different role. They participate to tissue and metabolic homeostasis, to wound repair, and they seem to be playing a role in cancer as well. So an easy question for us to ask was, in which of these different types of macrophages can we actually measure CD38? And so we can fairly easily differentiate a so-called progenitor macrophage into either an M1 or M2 using either LPS or IL-4. And then we can measure CD38 expression. And what we were able to show is that M1 macrophages actually are the ones where we can detect CD38. This is a quantitative PCR expression. We could do the same thing using flow cytometry. And we could do the same thing using an assay that allows us to measure NADA's activity. So we have a fluorescent substrate that gets degraded by CD38. And you can see here again that only the M1 macrophages have this activity, and it is CD38 dependent. If we look in a CD38 knockout mouse, we lose this activity. So the next question was, okay, knowing this the specific location of CD38 and expression in these cells, why and how is it activated? And so macrophages have the ability to become activated in response to a number of so-called molecular patterns. And there are two broad forms, the so-called pathogen-associated molecular pattern or damage-associated molecular pattern. The pathogen-associated molecular pattern, think about the monitoring of a host by the macrophages, which are part of the innate immune response, is to try to detect whether a bacteria or virus is actually present in the organism, even before this organism has started to replicate. And so the macrophages are responsive to a whole series of molecules that are shown on the left, on the bottom left, such as double-stranded RNA or LPS, which is a bacterial product. And what we saw is that, or single-stranded RNA as well. what we saw was that many of these signals, these PAMs, as we call them, actually were able to trigger CD38 expression, again, in an M1 type of response. And in contrast, none of the DAMs, the damage-associated molecular patterns, many of them such as amyloid, extracellular ATP, BSA palmitate, and so on, none of those actually were responsible, were able to activate CD38. So now you might wonder, what is the relevance of this to aging? And even though we haven't proven it, our hypothesis is that this might be linked to the progressive dysfunction of the intestinal barrier during aging. We're currently testing this hypothesis using antibiotic-treated mice. So what I'm showing you here is on the top is the lumen of the intestine, and underneath it is the intestinal, the epithelium from the intestine, then the lamina propria and the bloodstream. And we know that under normal condition, when there's no dysfunction, all of the bacteria and other products that are in the lumen of the intestine do not permeate this barrier. The barrier is tight. As a function of aging, we know also the function of the barrier decreases, and we know that some products actually are able to translocate. So think about LPS as a bacterial product, now is able to translocate in the lamina propria and eventually into the bloodstream, where it will meet macrophages. So we think, and we know a number of conditions where there is intestinal barrier dysfunction are associated with accelerated aging. And so one of the possibility, although not proven, is that this is one of the mechanisms by which CD38 expression increases during aging. The other way is, we think, is the mechanism that comes from senescent cells. And so I suspect that this audience has many people who know what senescent cells are. I'll just say a couple of words. So these are cells that accumulate during aging. They do accumulate in adipose tissue, and this is shown here. We can stain these senescent cells using beta-gal. And you can see, comparing a 12 months to an 18 months mouse in two different forms of adipose tissue, we can see a progressive increase in color. If we treat these mice with senolytic drugs, we can eliminate the staining as shown here on the right. Now, what is senescence, is a program. It's thought, we have Judy Campisi here at the back, who's been a pioneer in this field. The process of senescence is thought to be a protective mechanism against cancer. We know senescent cells accumulate during aging. And senescent cells, actually, interestingly, are characterized by what we call the SASP, a senescence-associated secretory phenotype, which is a series of pro-inflammatory factors that these cells secrete and that are thought to contribute to the chronic inflammation of aging. So the next question we tested is the possibility that these senescent cells might actually activate CD38 expression. And so the way we did this experiment initially was to take mouse embryo fibroblasts. This is a primary cells culture. We can treat them either with doxorubicin or with irradiation. We know both of these conditions induce senescence in this population. We can then harvest their supernatant and add them onto macrophages. And when Anthony Covarrubias did this experiment, he found that the SASP was a very strong inducer of CD38 expression, as you can see on the upper left panel, and much less so on CD157, and had no effect on the, at least no significant effect or weak effect on the other NAD consuming enzymes. So this really was an experiment that got us very excited, because it suggested for the first time a link between senescence, which we know occurs during aging, and a metabolic dysfunction associated with an NAD decrease via CD38. Now, the SASP, as I mentioned, is secreted by senescent cells. We do not know, and it's a very complex mixture of pro-inflammatory proteins. Birgit Schilling at the back and Judy Campisi published a paper a couple of years ago doing proteomics on the SASP, and we're able to identify hundreds of proteins. However, some of these proteins actually have been well characterized, such as IL-6, TNF-alpha, IL-10, some of the interferons, IL-12s. And so we tested these individually to see which one actually caused CD38 expression. And we identified 3 of them, IL-6, TNF-alpha, and IL-10. Now, I should add to this is that the fact that these molecules are doing it, are able to do it when we add them individually to macrophages, does not mean that they are responsible for this in the SASP. Of course, the relative abundance of each of these proteins in the SASP is going to be a critical determinant. And so we are currently testing the role of different cytokines using neutralizing antibody and using the SASP as a primary driver. So what I've told you so far is that we've identified 2 different mechanisms, the PAMs from presumably intestinal barrier dysfunction, and the SASP from senescent cells that both increase inflammation and increase CD38, leading to a decrease in NAD and to potential metabolic dysfunction or sirtuin dysfunction. So another question that was not clear and that I think we're providing a beginning of an answer is that I started by telling you that we had observed macrophage proliferation as a function of aging. And one of the things that really we wanted to reproduce, it tries to see, did the SASP do this as well? And indeed, when we, on the experiment that we did here was to measure proliferation, and we were able to show that indeed, using a flow cytometry assay based on EDU 488, we were able to show proliferation in the induced by the SASP, and this is quantified here at the bottom panel. So this really suggested that both activities from the SASP could be driving the processes that I've started with. There are a number of macrophage trophic factors in the SASP, including GM-CSF and CSF. So they could be responsible, but we haven't proven that they actually are doing it. So this adds a layer of complexity to this whole problem. The idea is not only inflammation, but is also macrophage proliferation during aging, which we think is driven by these cells. And those of you who work in the central nervous system have probably heard about gliosis and the proliferation of microglia that occurs in some manifestations of aging. And we think, although we haven't proven it, this could be driven by some of the same processes. Now, we were quite excited about all this data because it is something that is observed in multiple tissue. But then it was pointed to me that this actually was discovered close to 100 years ago by Elie Metchnikoff, who was a giant in the field of immunology. He's called the father of natural immunity. I show him here. He was working, he's originally Russian. He conducted most of his career at the Pasteur Institute, eventually got the Nobel Prize for discovering the macrophage. And he didn't not only discover the macrophage, he built a whole hypothesis about aging, claiming, because he noticed that these novel cells that he had identified, the macrophages and named, were also proliferating in multiple tissues as a function of aging. And so he had this whole hypothesis about the atrophy that one observes during aging, and posited that atrophy of organs in old age is very frequently due to the action of devouring cells. He called them macrophages because of their activity to eat other cells, which I have called macrophages. So really a remarkable finding. I think we're starting now by linking the activity of these macrophages to senescence. I think we're making significant progress in really understanding some of these long-standing mysteries. Metchnikoff, I called him a genius because that was not his only contribution. He also was the first person to study the microbiome and to raise the possibility that the microbiome might be playing a role in health. He searched the world for long-living humans, ended up in Bulgaria, and discovered some presumed long-living groups, a bit sort of the blue zone of the time, and realized that they were eating yogurt, and became convinced that from the yogurt work, became interested in the microbiome, essentially then brought yogurt back to France, and if you've ever had a yogurt called, I don't know if you sell yogurt called Danone in Chile, but I grew up in Belgium, and the major yogurt company is called Danone. That was founded in France based on the theories of Metchnikoff at the time. And I think we owe him probably to have this very broad distribution of yogurt in our population. And now what is recognized as an important way to maximize gut microbiome diversity. As you know, the whole microbiome field has just exploded way beyond what we could ever have imagined. And he created these two new fields. And I think this is quite inspiring. So, the result that I've showed you have been published, and I want to shift now to a couple of additional observations that we are currently pursuing and that are unpublished, and I think quite exciting. So one of the things that we did in the paper that was published was last December in Nature Metabolism, and the paper was focused not only on adipose tissue, but also on the liver. And I for the sake of clarity, and so I was not going to be bombarding you with too much data, I did not discuss anything about the liver, but there were some equally interesting things happening in the liver. When we were studying the liver, we found that senescent cells were slowly accumulating in the liver during aging. And this is illustrated here. If you do a beta-gal assay, you can find these blue cells progressively accumulating in the liver. And what we found also is that you all know that beta-gal is a marker, but it's not sufficient to determine whether there is increased senescence. There are other markers that we use that Judy has pioneered, including P16, and you can see here when we do mRNA transcript levels, we see increase in P16, increase in P21, and then also increase in inflammatory cytokines, IL-6 and IL-1 beta. So all of this painted a picture that the liver is also likely to be a site of increased senescence. Now, what was more perplexing was the fact that in what were these senescent cells in the liver? And so we conducted, actually in collaboration with the people at the BioHub, who generated a single cell analysis of liver, actually the whole mouse organs, to try to determine whether in which cells did the senescent markers appear. And so using this type of analysis and using the same markers that I showed you earlier, we found no markers of senescence on hepatocytes and on the endothelial cells, as shown on this figure. However, we found increased markers of senescence in Kupffer cells. And this is illustrated here. Again, you can look at, we found P21, CDK and 1A. We found other cyclin markers, HMGB1, which is a known, well-documented part of the SASP, IL-1 beta, and so on. And so this suggested that Kupffer cells might actually be senescent. And when we combined a marker for P21, again, which is a very, probably one of the more specific markers of senescence, we found it colocalized very strongly with F480, which is a marker for macrophages. And this is quantified here using an algorithm that we developed in the house. In the lab, you can see there was a much higher number of cells that were P21 positives and that actually co-stained for F480. So this was consistent with the possibility that a macrophage actually might become senescent. And for a number of reasons, the whole idea that macrophage might be senescent has been somewhat controversial in the field. There's been a lot of discussion. One of the reasons for this controversy is the fact that macrophages under basal condition express a high level of P16. And so it's been a little more difficult to, and remember P16 for many other cells is a marker of senescence, not for macrophage. They actually express a high level of P16 under basal condition. So the hypothesis became, well, maybe senescent macrophages represent a significant source of senescent cells during aging in the liver here, but maybe in other tissues as well. And so we tested this possibility by trying to induce macrophage senescence in vitro using, again, the same type of original macrophage that we can isolate. And we could either treat them with irradiation or doxorubicin. And you can see just simply by looking at their morphology, I mean, there's some changes that occur. Some of them are consistent with the induction of senescence, and I won't have the time to go into this. But when we look at these macrophages that have been irradiated, for example, we have the same data for doxorubicin. You can see they become almost uniformly beta-gal positive. Interestingly, they lose P16 expression. Remember, we've talked about P16 being a marker of senescence in most cells, not in macrophages. And discussing this disturbed me originally, but talking to Judy Campisi, she told me that there are no single marker of senescence in any cell type. Of course, in many cells, P16 and P21 are the markers, but there are many other places in which it doesn't work this way. So you did essentially define senescence by a collection of markers and the SASP. We indeed saw in these cells an increase in P21, and remarkably, we saw a very strong induction of CD38. So we believe that, and we have a whole series of other data that I won't have the time to go through, but I'll show you some further characterization of these cells. So this data on CD38 triggered our interest because it suggested that senescent cells can not only induce CD38 expression in other cells via the SASP, but they can also sometimes induce it in themselves, as in the case of macrophages. So we've spent quite a bit of time in the past year with Anthony, who has now moved on to become a junior faculty member at UCLA. We continue to collaborate to finish the story together, to do proteomics analysis to look at the SASP from these cells. So we took the secretome from macrophages and senescent macrophages, and we're able to show a large number of proteins that become secreted by these senescent macrophages. So this is irradiated macrophages. We see the same thing after doxorubicin treated macrophages. When we compare this population and this population, we find a very strong overlap. So even though doxorubicin and irradiation are two different stimuli, they trigger a very similar response in macrophages. So the ongoing work that is going in this field is that we are studying senescence also in human macrophages. We can reproduce these data in macrophages that we can isolate from the blood. We are conducting proteomics, transcriptomics, and metabolomics analysis of these senescent macrophages. We're also pursuing the identification of macrophage-specific senolytics and xenomorphic agents. Now, how do we link the story that I've showed you so far with this macrophage senescence? Is that we believe that one of the ways in which these macrophages become senescent during aging is via their increased proliferation. I've showed you our data and the data from, I haven't shown you the data from Mechnikov, but I've told you that he's the one who originally discovered this, that we believe that during the lifespan, the resident macrophages are under a proliferative pressure, which increases the function of aging. And we know there are multiple ways to become senescent. One of them is via replicative senescence. This is the idea that most primary cells have a finite replicative lifespan. And remember that these tissue macrophages that are resident are not renewed via stem cells. They are deposited early during life. And we believe that a hypothesis that we will be testing is that as they replicate throughout life, a fraction of them adopts this replicative senescence type of phenotype. And so the summary here is essentially what we think is a working model. Of course, there are many questions that remain to be answered, but we're continuing to characterize this and trying to understand really this complex relationship between senescence and CD38. Now, I want to finish by sort of discussing maybe what these observations have actually highlighted in terms of therapeutic potential. And I've told you about, we've already gone through this whole mechanism, NAD being pulled by the PARPs and by CD38, the recycling pathway that leads to, from CD38, leading to nicotinamide and back to NAD. This is so-called salvage pathway. So the field has been busy trying to essentially activate the sirtuins. And I'm sure you've heard about the work of David Sinclair on resveratrol and the so-called stacks, which are sirtuin activating compounds, which were generated by a company called Sirtris. You probably know also that many of these actually have failed in the clinic. And we believe that one of the reasons for the failure is the fact that that's not the primary problem during aging. And if you can activate these enzymes, but if NAD is limiting as it is in older people or in select diseases, you will not get the desired effect. And I think in the future, there's going to be a lot of interest in combining some of these stacks or resveratrol with some of the other agents that modify NAD abundance. So there's been an interest by a number of colleagues, including Johan Aurix in Switzerland, to inhibit the PARPs with the idea that this might actually also lead to elevated NAD. The problem, this works actually in C. elegans because it lives on average for 21 days. The problem if you inhibit the PARP, so the PARP inhibitors in C. elegans increase lifespan and raise NAD levels. The problem if you do this in long-lived animals is you interfere with DNA damage repair and you're creating a whole series of other problems. So I don't believe there's any possibility of using these inhibitors for a lifespan intervention. There are some efforts to try to activate NAMPT. There's some evidence that NAMPT, which is a key enzyme in this whole salvage pathway, becomes rate limiting during aging. That data has become a bit controversial, but we're still working on this area as well. And finally, you've all heard about some of the precursors, nicotinamide riboside or nicotinamide mononucleotide, which are two precursors to NAD synthesis, in part because they are bypassing the enzyme NAMPT. And there's interesting data that suggests that some of those might have protective effect against some of the manifestation of aging. And we have a program in the lab working on nicotinamide mononucleotide. And finally, CD38 becoming overactivated during aging offers a novel therapeutic target. For full disclosure, we started a company called Napa Therapeutics, which is exploring the development of a small molecule inhibitor for CD38. And we have these inhibitors, and we will be testing them shortly in a number of indications, including during aging. So what I want to finish now is to sort of highlight what I've told you today is that starting from deregulated nutrient sensing, we went back to NAD right here, and then made us realize that there was a link between NAD and cellular senescence. There's also a link between stem cell exhaustion. There's a link between NAD and epigenetics and genomic instability via the PARPs. So this is obviously a picture that is still quite complex, but I think it is really helping us in trying to bring a network view of aging and hopefully increase our ability to intervene against it. I want to finish by acknowledging the key people in the lab who have worked on this, in particular, Anthony Covarrubias. This was primarily his work. Rosalba Perón is a scientist in the lab who's been doing a lot of work also with Anthony on NAD metabolism, and now is in charge of our effort with Napa Therapeutic. Birgit Schilling and her colleagues is a big proteomics lab at the Buck. We've collaborated with her over the years. She's been instrumental in many of the observations that I've showed you today. And finally, I just want to particularly highlight the collaboration with Judy Campisi. When I came to the Buck, I think this became what I would call a marriage made in heaven. I think it's been wonderful to collaborate with Judy on senescence and to continue building on this story. And I will stop here, and I thank you all for your attention, and I'll really be looking forward to your questions.